Hi everyone, in this screencast we are going to talk about anaerobic respiration. So if you recall from class, obviously before we can do either aerobic or anaerobic respiration, we will go through glycolysis. And if you recall, in glycolysis we're going to take a glucose molecule and then split it into two 3-carbon pyruvates. Obviously I'm not showing all of the steps there, but remember that it has a net yield of 2 ATP per glucose molecule. It also is going to create 2 NADHs per glucose, and those are going to be important and sent to the electron transport chain in the mitochondria, which I'll discuss later on. So then if oxygen is not present, those 2 pyruvates are going to then be converted to lactic acid, or they can be converted into obviously ethanol as well, using fermentation or the process of anaerobic respiration. But if oxygen is present, we are then going to do either the citric acid cycle, or it's also known as the Krebs cycle. So a couple of key points about this before I go into the details of it. First is, where is this happening? You guys know generally that it's happening in the mitochondria, but specifically it's going to be happening in what we call the inner mitochondrial matrix. On the next slide, I'll show you a picture of what this looks like. But just a couple of other things before we get there to give you an overall picture. Obviously, this is going to be an aerobic process, and it will only proceed in the presence of oxygen. Also, if we're thinking about our net yield, remember those are important things to keep in mind when we're going through the process of cellular respiration. We are going to be creating 2 ATP molecules per glucose molecule. Then we can also look at our net yield of NADHs and FADH2s. We are going to make a total of 6 NADHs and 2 FADH2s. So the process of this is what will happen here. More detail on this later, but the NAD plus will be reduced to NADH and the FAD plus will be reduced to FADH. So in general, in this stage of cellular respiration, the oxidation of glucose to CO2 is going to be completed. So keep all of these little bits and pieces in mind as I go into a little bit more detail now. So here again, our summary of what happens with glycolysis. And we can see those 2 pyruvates, obviously they are 3 carbon molecules, hence the 3 black dots. Then we're going to take those pyruvates and do something before called the preparatory steps, or the PrEP steps for short. So we take those 2 pyruvates, and then we're going to add 2 of something called coenzyme A. Don't worry about what that is per se, just know that that is being added. Then we also are going to add 2 NAD pluses. And then putting all of those things together, we're going to create 3 things. First, we're going to create 2 things called acetyl coenzyme A. Then we're going to create 2 CO2s, and then lastly 2 NADHs. So I've gone in and drawn in the carbons for you so you know how many carbons we have in each of those examples and things that we create. So this acetyl-CoA that we've created, this is going to be really important because this is what we'll put into the Krebs cycle. So if I had more room like you, I would just sort of scroll down and then draw beneath it here, but the slide is done, so I need to move on to the next. So I'll copy the preparatory steps, those PrEP steps, to the top so you know that, again, this is coming after it, but for your notes, you should just continue right under those PrEP steps. There are the PrEP steps. Let's use the acetyl coenzyme A, or short for acetyl-CoA. So that acetyl-CoA, the first thing it needs to do is combine with something that you have heard before, and that is something called oxaloacetate. And you'll remember that it is a 4-carbon molecule. So combining those 4 carbons with the 2 carbons of acetyl-CoA, and that of course will give us a 6-carbon molecule. You don't need to know what this is called. You can just refer to it as the 6-carbon molecule. We're going to do 2 things to it. First, we'll add in NAD+, and from that we will make our NADH. We're also here going to make some CO2. And then from that, obviously we've taken 1 carbon off, so we're left with only 5. The same thing will happen to those 5 carbon molecules. We add in the NAD plus and we get out the NADH, and we also are going to take another carbon off in the form of CO2, and then from there we're left with a 4 carbon molecule. So we're left with that 4 carbon molecule, and then we're going to do a bunch of things to bring it back to what is oxaloacetate. First, we're going to add in ADP and also a phosphate. Remember, we put the little I after it. It just stands for inorganic. You can just write it down as P for phosphate if you'd like. That will then make 1 molecule of ATP. Also, we are going to need to make some FADH2, so we do that by putting in FAD, creates the FADH2, and we're also going to create some more NADH, and we do that by adding in NAD+. And doing all of those things will bring us back to create that oxaloacetate. But you need to keep in mind that we have 2 of these acetyl-CoAs, so that means that we're going to have to do this cycle twice. We have to go through it 2 times, and so everything that we create here in this entire Krebs cycle, we're going to make double that what we see here. So let me just highlight some of those key things. Obviously creating NADH is good, FADH2 is going to be good, the NADHs, all of those are highlighted to remind you that those are important. We can use those to make more ATP, which I'll describe later on. And here, of course, is the ATP that we are creating. Obviously, going through this whole process, this whole cycle, we're only creating 1 ATP, but per 1 glucose molecule, obviously, since we do this cycle twice, we're going to create only 2 ATP. So there we have the Krebs cycle for you. And in the next screencast, I'll tell you guys about what we then do with our NADHs and our FADH2s to make more ATP. Okay, that ends the screencast here on the Krebs cycle. Again, just to put this all into perspective for you, remember that we are going to start with glycolysis. That doesn't require oxygen. Then if you don't have oxygen, you can do anaerobic respiration. Or if you do have oxygen present, you can do aerobic respiration. From there, we're going to do the prep steps first. That will create our acetyl-CoA for us, which we then need to do the Krebs cycle, also known as the citric acid cycle. And after, I'll tell you guys in the next screencast about something called the electron transport chain. But this is where we're ending here after the Krebs cycle. So there, also add in the prep steps part. Okay, have a good night.